I just want to check my yes, okay, fine. There we go. Hello, everybody.
Hello.
Hey, everyone. Hi, nice to see you all. Today we'll be doing a live class every Wednesday, okay? Unless you have a quiz on Wednesday, in which case we'll shift it to the Monday. In addition, don't forget that tomorrow at 1 o'clock, I have a question time for half an hour, okay? And you should already have that meeting ID, but I can send it out again if you don't, so to make sure that everybody has it. Let me just see how many.
Sorry,
we've got
can you
18
repeat?
people in the room. Hello?
Sorry,
Go
can you
ahead.
repeat the times for tomorrow that you said?
Yes, it's at 1 o'clock. So I'll send you an email. I'll send you an email to remind you all that it's there. And I'd say a half an hour, but if there are lots of questions, we'll go more than half an hour. I'll be there for half an hour. If nobody comes after half an hour, I won't be there. But if you want to stay longer, then that will also be all right. Okay, everybody. So the first thing to say, I want to make sure everybody can see my screen correctly. Can you see the shared screen saying microbial growth? Somebody
Yes,
say
yes.
Yes. Yes.
Okay, fine. Thank you. So today we're going to be talking about microbial growth, and I strongly suspect that this will go over 2 classes because it's quite a long chapter. We've been talking so far, we talked intensely about the metabolic processes, and we saw how prokaryotes take matter and energy and how they deal with it to build biomolecules and then to build the cell and the structures of the cell. So today we're going to be talking about microbial growth itself, both in terms of the growth in size of the cell, but then particularly about the growth of populations of bacterial populations. So in order for growth to take place at all, there are certain requirements which prokaryotes have. First of all, they have to have the correct physical requirements. But in this, I must tell you, there's an enormous diversity of prokaryotes. They can occupy virtually every habitat. And some of those habitats are extraordinary. It just depends what those prokaryotes are adapted to. So they may be adapted to living in areas with high acid where no other bacterium could live. They may be adapted to living in high temperatures where no other cells could possibly live. So the bacteria are very diverse, and they've diversified to occupy many, many, many ecological niches. But for each of them, for each of those habitats, the cells occupying them will have specific requirements. And they'll have specific requirements, especially for temperature, pH, and then for the correct osmotic pressure, the correct amount of solutes outside the cell. We'll see how this works in a minute. Then in addition, that applies to us as well, of course. We have to have an optimal temperature, pH, osmotic pressure, et cetera, but we are able to regulate a lot more than something like a prokaryote is. Prokaryotes are entirely dependent on the environment. They can do very little to control the physical nature of their environment. In addition, living things require certain chemical compounds. We have to have matter to build our biomass. Prokaryotes are no different. We have a requirement for carbon, first of all. Then for especially the other atoms which are in high demand in living systems are nitrogen, sulfur, and phosphorus. All of those we'll discuss in a minute as to where they go and why. Then there's a requirement for very small amounts, mostly of metals. And some of them sound very strange, some of them not so strange. Iron, for example, you'll know we have iron in our blood, in our heme groups of our blood. But some enzymes especially require the presence of trace, very, very small trace amounts of things like molybdenum and copper and these sort of things that they operate with. The next really important thing, which we'll talk about at some length, is the amount of oxygen in the environment. And this is critically important. And just to start off your thinking, we sort of automatically think, well, oxygen is good for you, right? It's you breathe it in for respiration and everything. But I think that I told you when we talked about cellular respiration, that in fact, oxygen is a very dangerous molecule. It can actually cause a lot of damage to biological molecules. It is very, very reactive, and it occurs in different forms, some of which are more reactive than others. Nonetheless, when we look at biological systems, we see that some cells have a positive requirement for oxygen. Others have a requirement that there be no oxygen, and some of them can switch between the two. We'll see that in a minute. The last thing that they require, we'd refer to as organic growth factors, things like vitamins and this sort of thing. Actually, bacteria are very, very good at making these themselves. For the most part, they don't require much from outside, but if they do, which some of them do, we have to actually supply organic growth factors. Also, very often they form part of the enzyme systems. But if we have to supply them, we say that they are essential growth factors. They're things which a cell cannot make itself. It has to get them from an outside source. So let's have a look first at the role of temperature and of environmental temperature, remembering that bacteria prokaryotes are entirely at the mercy of the environment for determining what their temperature is. They have no way of controlling their temperature. So you'll see here, just looking through this graph here, you'll see that there's an enormous range of temperatures. Each of these curves is a growth curve. There's an enormous range of temperatures over which prokaryotes can live. Look here, all the way from close to -10, all the way up to 110. That's way above boiling. So depending on the prokaryote that you're talking about, they will be adapted to a particular range of temperatures. We'll take this group here in the middle just to start off with, because this is by far the largest number of prokaryotes are in this group of called the mesophiles. And almost all our pathogens are mesophiles. But we'll use it to illustrate, and you can see here, this is a growth curve. So it's growing best where the curve is highest. So it's growing best here, and that we call the optimum. That's the optimum temperature. And in this case, for the mesophiles, it's right around 37 degrees Celsius, which, not coincidentally, is our body temperature. So most of the bacteria associated with us are mesophiles, and they are adapted to our body temperature. That's where they grow best. Many, many environmental bacteria also will be in this range somewhere. They'll be, if their optimum might be, if you're looking at an individual bacterium, its optimum might be a little bit different. But for most environmental bacteria, their optimum is around about here, and they are also mesophiles. That's the optimum. Then there is a minimum growth temperature. In the case of the mesophiles, it's set at about 10 degrees Celsius. So that is cold, but it's not freezing. This is kind of the temperature of getting down to where your refrigerator, actually your refrigerator is a little bit colder, but in this range. And at that temperature, growth stops. The bacterium is not able to reproduce. And that's one of the reasons why refrigeration works. Because we actually take, in refrigeration, we take it down, the temperature down to here, way below the minimum growth temperature for most mesophiles. They cannot grow at all. If you raise the temperature again, then they will begin to grow again. So that is the minimum growth temperature. But there's also a maximum. Here's the optimum. And if I slowly increase the temperatures on that, the x-axis, if I slowly increase the temperature, you'll see the growth efficiency drops quite rapidly until it stops altogether. But this is a little bit different. This is the maximum temperature, maximum growth temperature. This is a little bit different to the minimum in that if I take bacteria and I keep them at this temperature for any length of time, and I then cool them down, I'll find that most commonly they will not start growing again. The reason for this is that as we raise the temperature, we begin to damage the proteins. We begin to damage especially the enzymes that are driving the metabolism. And we actually end up irreparably damaging the proteins, and that is called denaturation. So we denature these proteins, and we cannot, once they're denatured, we cannot restore their function. So maximum growth temperature is usually lethal to take them above that. Minimum growth temperature, it's seldom lethal to take them below. For some of them that are fussy, it can be, but that's not common. Usually they survive perfectly well at low temperatures, and they will begin again, growth again when you raise the temperature. This is our most important group. These are the mesophiles. So have a look now at these ones here, because these also cause us some problems. These are called the psychrotrophs. If you look at their optimum growth temperature, it's above 20. That's quite warm. So their optimum is way below that of the mesophiles, but it's still not in cold. But they are cold tolerant. Look, they will continue growing well below their optimum. Here's around about here is where our refrigerators are. And you see that they are still growing. They're growing slowly, but they're still growing. They are cold tolerant. That's why they're called psychrotrophs. They're able to survive at cold temperatures. They stop growing down at freezing, around about freezing. And they grow quite happily to above 30 degrees Celsius. The reason I say that these can cause us problems is because there are some food poisoning organisms which will grow in the refrigerator. And there's one in particular called Listeria. And Listeria causes quite a dangerous food poisoning. And one of the big problems of dealing with Listeria is that it will grow down here. It will grow in the refrigerator, even if you've got something contaminated, food stuff contaminated with Listeria. You put it in the fridge thinking the fridge is going to keep it safe for you. Listeria is not a mesophile. If it was a mesophile, that would be fine. Growth would stop, but it's not. It's a psychotroph. Growth continues. It continues slowly, but nonetheless, as we're going to learn later on, when you're dealing with exponential growth, when you're dealing with a population that doubles its size at regular intervals, it's not long for any of these things before you build up a very high population number. So that's a problem for things like Listeria. These bacteria here, there are no pathogens down here. These are called the psychrophiles, and these have a positive requirement for cold in order to grow. Look at their optimum here, and you see it's just about 11 degrees Celsius. That's cold. And this is typical for marine bacteria, for many marine bacteria, for example. They grow in cold temperatures. Their optimum is quite low. And they can continue growing in very cold waters. That would be typical of Antarctic waters, that sort of thing, ice ponds. All of these places have their own populations of bacteria which are adapted to this cold. So the next group we'll talk about are these, and they're called the thermophiles. That means they like high temperatures, and they have a positive requirement for high temperature. Have a look here. Here's the optimum, and it's above 60. 60 degrees Celsius, if you put your hand into 60 degrees Celsius, it feels like you are burning. It's that hot. And yet these are able to survive quite well. In fact, they have a requirement for high temperatures. And their maximum growth temperature is way up there. It's over 70, 72, somewhere around there. And the minimum is very high. The minimum is 40 degrees Celsius. That's already very warm, and they stop there. So these guys are especially adapted to hot water. Excuse me. Now you've got to ask yourself, where would we find that? Where would we find hot water like that? Well, the answer is you'll find it in places like Yellowstone National Park and at volcanic springs. And these natural hot springs are places where these bacteria, the thermophiles, can be found. And as microbiologists, we might not often encounter these things, but we are particularly interested because if we culture these bacteria, the thermophiles, we can extract some very, very useful enzymes from them. And one of them in particular, you're going to hear about later on, and it's a DNA polymerase, an enzyme that replicates DNA. And it is specially adapted to work at high temperatures. And we find that very, very useful in a particular experimental stratagem called polymerase chain reactions. We use this enzyme in that particular reaction. But these are not things you're going to encounter in a normal environment. These are not things you're going to encounter as pathogens or anything like that, or even in any environment really that we normally would deal with. I will tell you they have been extracted from hot water cylinders and hot water taps and things, but they're not pathogenic or anything. So those are pretty extraordinary. They must have some very, very specially adapted enzymes. But look at these ones. These are the hyperthermophiles. And look where their optimum is. It's right about 94, 95 degrees Celsius, very, very close to boiling water. And they've survived quite happily and continue growing at boiling temperatures. These are the only living cells which can live at these extraordinarily high temperatures. They have a positive requirement for these hot temperatures. You can see that because look where their optimum is. It's right up 94, 95 degrees Celsius. Where do these occur? The answer to that is they occur especially in very, very deep marine systems where there is volcanic activity. And the pressure of the water is so great that it stops the water from boiling. If the water doesn't actually boil, it is well above boiling temperature, which is 100 degrees Celsius at sea level. It goes up to about 115 degrees Celsius. But these are adapted and they will carry on growing well above the boiling point of water. And that's their special. And these are submarine vents, submarine volcanic vents. And these bacteria actually form the basis of the whole ecosystem there. The whole ecosystem is where our ecosystems are based on plants, for example. These ecosystems are based on these bacteria, which are quite happy at these temperatures. Okay, so be sure that you know these 5 groups of the different bacteria, of temperature requirements. Here, this just illustrates for you. These the consideration of growth rates and temperature becomes very important for us when we think about managing foodstuffs, for example. I'll give you a simple example. We have on our skin a very, very common bacterium is Streptococcus pyogenes, and and another one called Streptococcus aureus. Staphylococcus aureus. These bacteria can grow quite happily away from the body. Now, Staph aureus is really interesting because we commonly produce a Staph aureus if we have a cut or something. It gets infected. We will actually grow Staph aureus quite vigorously in the cut. We might not even really notice that we've got the cut. And you have been invited to a picnic, and you make a potato salad, and potato salad's full of good stuff for bacteria. It's got your mayonnaise, eggs, oil, all sorts of things in there where bacteria will grow very, very well. And you got scratched by your cat 3 days ago, and you've got a tiny little sore on your fingers. You don't even realize that it's full of Staph aureus. And you mix up your potato salad and you take it and it sits out there on the table for just an hour or so is enough. The Staph aureus takes off because the temperature rises here. It's a mesophile. And the temperature rises to around about between 40 and 50. It's right there where the mesophiles are still doing really, really well, and they grow really rapidly. This is a very dangerous temperature range because it's the kind of temperature range that our food is subjected to if we simply leave it in the normal environment. This is a kind of temperature at which mesophiles will grow exceptionally well, and that is what Staph aureus does. It grows rapidly inside your potato salad. And when it does so, it releases toxins. And when you eat the potato salad, very quickly, within a half an hour or so, you have a violent reaction to it. This is the typical food poisoning situation, where somebody has contaminated food, left the food at around about this temperature, and the bacteria grow and intoxicate the food. They put toxins into the food, which cause violent diarrhea, violent vomiting, that sort of thing. This is the sort of thing when suddenly 3/4 of the people are rushing off to try and get home or to the nearest loo. So the answer to that is, even if you did contaminate it, if you kept that mesophile, Staph aureus, at a low temperature down here somewhere, they might survive, but they will grow very, very slowly. And your food will actually stay quite safe. You might not even notice that it was ever contaminated. If you then warm it up again, leave it out again, you could again be in trouble. The problem that comes down here around about in refrigerator temperatures. As I told you, there are bacteria like Listeria, which can also cause food poisonings, which will survive down here. There are not a lot that would do that, but there are some really significant ones. So these are all the kind of environmental temperatures we might subject our food to. What about above that? Well, above that, above 60, there are not many mesophiles, or not many bacteria that we would normally encounter, which will be very happy above 60 degrees Celsius. This was discovered by Pasteur. You remember we talked a lot about Pasteur in the history of microbiology. He discovered that if he raised the temperature especially of liquid, above 60 to about 65, and he kept it there for a good length of time, he would then let it cool, and then he would do it again. He found he could kill off most mesophiles. He could kill off most of the bacteria that might infect that. And he didn't sterilize it. But what he did do was he found he could eliminate especially the pathogens. This was very important for milk, because milk, in the old days, milk was actually quite a dangerous substance, because it's a very good bacteriological medium. Bacteria grow very well in it. And it could grow a variety of human pathogens, could grow in milk. And there were regular incidents of mass infections and things that arose from people drinking contaminated milk. But Pasteur discovered if you raised the temperature of the milk, kept it there for a good length of time, cooled it, and then repeated the process a couple of times, you could eliminate most of the pathogens, and the milk became much, 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 much safer to drink. And that is the process of pasteurization. That's where the word comes from. We say it's like an orange juice is pasteurized. It's named after Pasteur, who invented the process. Above this temperature, up anywhere from there on up, something interesting happens, because in fact we can kill off most active cells. Most vegetative cells can be killed by heat. But there's a problem, there's a big problem, and it's a problem with pasteurization as well. And that is that it does not kill off bacterial spores. Bacterial spores produced by Clostridium and Bacillus are very, very resistant to these high temperatures. They don't start growing at those high temperatures. It just doesn't affect them. And when you take those spores back down to a reasonable temperature and give them the correct growth habits and everything, they will germinate. The spores will germinate quite happily. Now that is very dangerous, because it means it becomes very difficult to pasteurize or heat kill spores. And you can't sterilize, you can't be sure that something is sterile just by heating it. Instead, we have to resort to putting stuff into an environment where we raise the pressure. We raise the pressure so that we can raise the temperature of substances well above boiling point without it actually boiling. And that is the system that we use. It's what we use in the laboratory. It's universally used for sterilizing under most circumstances, and that is the system of autoclaving. And autoclaving takes the temperature way up here, way above boiling point, and that will kill the spores. And you can be sure if you've autoclaved at proper conditions for the proper length of time, you can be sure that you have actually sterilized whatever it is that you're dealing with. Is everyone okay?
So
No
far, so
question.
good.
If you have, okay.
Yeah.
If you have questions, okay. If you have questions, you just unmute and shout, and you will appear at the top of the list. Okay, so that's temperature. We see that they have an optimum, maximum, and a minimum. A similar system applies to pH. Proteins are extremely sensitive to pH, and the reason for that is because their secondary and tertiary and quaternary structures are all dependent on these weak bonding systems like hydrogen bonding, hydrophobic bonding. All of those systems of weak bonding are very important in structuring proteins. And if you start changing the pH, you radically affect things like the hydrogen bonds. You cause them to break, and the structure of the protein begins to be lost. And if you disturb the structure of a protein, you will radically disturb its function. So most of our enzymes, 99% of them, they are proteins, and the enzyme function is intimately tied to that structure, to the structure of the protein. If I change the pH, I start changing the shape of the protein, and it will lose its function very quickly. Sometimes it can be restored by restoring the pH, correct pH, but many times changes in pH can actually denature a protein. So the pH range is critical for enzyme function. We'll find, unsurprisingly, I suppose, that the vast majority of bacteria, and in fact, the vast majority of cells, operate at around neutral, around about 7. They sometimes can operate quite happily a little bit above, a little bit below 7, but it's usually around 7 that the optimum lies. And those we call the neurophiles, and that's by far the vast majority of living systems operate around there. There is a minimum pH, and look where it is. 5.5 is where the minimum pH for growth for the neurophiles is. That's not very low. That's really not very low at all. Similarly, here, the maximum at about 8.5 is not very alkali. So this is quite a narrow range, and it's quite a narrow range. And we'll find that many living systems buffer their interior. So in the inside of the cell, there are chemical systems which buffer the pH. They prevent the pH from moving higher or lower. And especially, they prevent the pH from moving away from the optimum rapidly. So that is the function of physiological buffers inside the cell, is to maintain the optimal range. Just let me try something here. There we go. Okay, so there are some bacteria which are adapted to strange habitats. Honestly, bacteria, prokaryotes, are so diverse that really you can pick any habitat on the planet, and you can be pretty sure there's going to be some bacteria there, no matter how extraordinary it is. These ones here are called the acidophiles, and look at their optimum pH. It's way down here at 3, and they will go even lower than that. They're still growing quite happily around about here. These are the acidophiles. Now, in nature, we'll find acidophiles in environments where the water is naturally acidic. And the one that springs to mind immediately is acid mine drainage. This is a huge problem, environmental problem, which we won't go into why the water is acid, but the water draining out of many coal mines, for example, comes out at extremely acid. And the pH is way down 2 or 3. But there are bacteria which will grow quite happily there. And they are specially adapted. Their proteins and everything are specially adapted for those low pHs. In fact, they require a low pH to grow properly at their optimum. Likewise, there are some bacteria, some prokaryotes, I'd say, not necessarily bacteria, which are alkaliphiles, and they grow at a high pH. Their optimum is a high pH there. Again, for some extraordinary environmental situations, like hot springs, volcanic ponds, these kinds of things may in fact be very alkaline, and they also have their own bacteria. For our purposes, these are the ones that we are most interested in, the neutrophiles. Okay, the next thing to think about is the osmotic environment, is what is the concentration of salts outside of the cell. Remembering all the things we've said about the cell membrane, that the cell membrane is semi-permeable. It does not allow the passage of ions very fast, very quickly, but it does allow the passage of water across the membrane. And realizing that, we've discussed before, if we take a cell and we put it into an environment where the outside, there is very few solutes in the environment. It's very dilute around the cell. Well, the inside of the cell is very concentrated. And under those circumstances, water will flood into the cell due to osmosis. Osmosis is a diffusive process whereby water moves from an area of high water concentration to an area of low water concentration. High water concentration outside, if there's very few solutes. Low water concentration inside the cell, because there is little water compared to how many solutes there are. Water moves in. In many systems, this is extremely dangerous because the cell will begin to swell. And this is the primary function, we said before, of the cell wall, because the cell now will swell up against the cell wall. And when the force exerted by the cell wall is the same as the force of the membrane pressing out due to osmotic pressure, then the net inward passage of water stops. The cell stops swelling. As much water passes in, passes out. It's an equilibrium situation. So that is the normal situation for bacteria. They actually usually prefer an environment which is slightly hypotonic. And that is also, I'll just tell you, the case for plants. Plants positively require a hypotonic environment so that their cells swell strongly against the cell wall. Bacteria are quite happy in an isotonic solution as well. Plants are not so happy. Bacteria are quite happy in an isotonic. That is when the amount of salt inside is the same as the amount outside. You can think of it the other way around. An isotonic situation is one where the water concentration is the same inside the cell as it is outside. Water is still passing back and forward across the membrane. But in an isotonic situation, or where in a hypotonic situation where the cell is fully swelled against the cell wall, as much water passes in. If a molecule of water passes in, a molecule of water passes out. And the cell is quite happy with that situation. There's a great danger for bacteria and for plant cells as well, or basically anything. If you dump a cell into a hypertonic solution, that is, into a solution where the salts outside are very much more concentrated than they are inside. Now we have the reverse situation, and it's this, that if the solute concentration out here is very high, and it's higher than it is in here, then the water concentration is higher here inside the cell than it is outside. And water will flood out of the cell if it attempts by diffusion trying to even up the concentration of water on both sides. It floods out, and the cell now shrinks. It collapses into the center, away from the cell wall. And this is a situation called plasmolysis. And it's very dangerous because if it proceeds, then all of the cellular structure starts to get disrupted. And the cell often will not recover. Sometimes they will be able to recover if you restore them quickly, but very often plasmolysis like this is lethal. So that's a bad situation. But you'll realize it's not a situation that arises all that often. There's one situation for us where it does arise, and that is on our skin, because we produce salt in our sweat. And we can end up creating this kind of environment which will kill off many pathogens which land on our skin. That is why salt in our sweat is a good disinfectant. But many of our bacteria, I should tell you, are adapted to that high concentration. And the bacterium I already told you about, Staphylococcus aureus and Streptococcus pyogenes, some of these bacteria are adapted to these high salt concentrations, and they're quite happily. They manage perfectly well. But this is the principle that we use for preserving food. This is why we salt meat, for example. This is why we salt fish to preserve it. This is also a system which is used in pickling vegetables and this sort of thing. What we're doing in those situations is we're creating an environment which is very, very hostile to the growth of bacteria. Most bacteria there will actually plasmolyze and die because the salt concentration is so high. That's why we have learned through all of the millennia how to salt food to preserve it. Okay, so let's just think also about some of the actual requirements of matter that a cell will have. And we said before, we listed these, but now let's try and understand why cells need these things. The first is carbon, of course. It's our primary requirement, is we need carbon because it's the structural backbone of all of our molecules. All our organic molecules are built on a skeleton of carbon because of carbon's versatility, the versatility of the way it's able to make different bonds and things like that. We are chemoheterotrophs. That means that we are absolutely reliant on already created organic molecules. And we're reliant on them for both the matter they contain, for the carbon they contain, and other things. But we're also reliant on them for the energy contained in their chemical bonds. Energy contained in the chemical bonds is potential energy, which we can transform to use as our cells see fit. We need to remember, though, that there are those organisms, like photosynthetic bacteria, for example, which are able to use carbon dioxide as their carbon source. And these we call the autotrophs. We are heterotrophs, and we are chemoheterotrophs. We depend on chemicals already made. The autotrophic organisms, such as photosynthetic bacteria, and those strange groups of bacteria like the chemosynthetic bacteria, are autotrophs, and they use carbon dioxide as their source of carbon. The next thing, probably the next most important, they're all important, but if we were to rank them, the next most abundant in our molecules is nitrogen. And that is because nitrogen is a component of so many different things, so many different organic molecules contain some nitrogen. First of all, of course, the proteins, which have a whole amino group. On every amino acid has an amino group associated with it. So proteins contain a lot of nitrogen. DNA, the nucleic acids, also contain nitrogen. All of the nucleotides contain nitrogen, and that means that that very important nucleotide, ATP has a nitrogenous group associated with it, a double ring called adenine, and that's where it gets its name, adenosine triphosphate. It's a nucleotide, and it contains a lot of nitrogen. Now, the sources of nitrogen are generally, for the most part, the bacteria will enzymatically digest substances which already contain nitrogen. They will break down proteins, for example, to use the amino acids, or even to break down the amino acids to use the nitrogen for themselves. So they're generally depending on a source which already contains a lot of nitrogen, and they enzymatically break it up, hydrolyze it in order to use the nitrogen. Some bacteria are able to use an inorganic source of nitrogen, like ammonium or nitrate. And I'm sorry, I thought I'd change this. That there needs a correction. It should say inorganic. Some bacteria use ammonium nitrate from inorganic material. They're actually able to utilize those inorganic forms as a source of nitrogen. And then there are some tremendously important, ecologically tremendously important. There are bacteria, there are not a lot of them, that have extraordinary enzymatic methods to fix nitrogen, to fix gaseous nitrogen, N2. N2 is practically inert. It behaves almost like a noble gas. There is almost nothing that it reacts with. But some bacteria have developed systems to fix nitrogen, to convert it from atmospheric nitrogen into first ammonium, and then into various other nitrogen species, and then into amino acids. These bacteria are the bacteria that are associated, for example, with the legumes, with peas and beans and all of those plants. They have bacteria in their roots which are able to do this. And then, excuse me, there are these other bacteria which are using these substances, ammonium and nitrate and those sort of things. These all nest together. Unfortunately, we're not going to have a chance to talk about the importance of environmental bacteria in cycling of minerals and nutrients and everything else. But these bacteria are tremendously important in the cycling of nitrogen in the biosphere. And just think of it this way. We are absolutely, most things on the planet are absolutely dependent on eating their nitrogen source. They have to take it in the form of protein or whatever, and then break those down. That's what most of us, including the prokaryotes, most prokaryotes and us, that's how we operate. But the problem with that is that eventually, those compounds are broken down completely, and the nitrogen is lost as waste products, and ultimately is lost as gaseous nitrogen. And it's lost as gaseous nitrogen because of things like this. And we are absolutely dependent on these bacteria here to continually fix nitrogen and feed it back into the ecosystems in a form that we can all use. The nitrogen, the processing of nitrogen through ecosystems is very, very, very complex. All that you need to remember is that complexity is dependent on having bacteria that can manipulate the nitrogen. Bacteria also, prokaryotes also have a requirement for sulfur, and that is because it forms part of a couple of very important amino acids, and in particular one called cysteine and then another called methionine. They contain sulfur. Cysteine is very important in proteins because two cysteines near one another can use their sulfurs to bond to one another, and they bond with a very strong bond between the sulfurs called a disulfide bond. And it's a very important way of structuring proteins. That is a tertiary feature of protein structure. Sulfur also occurs in many cofactors, many organic cofactors, thiamine, all those sorts of things. Most bacteria will easily obtain sulfur by breaking down amino acids like cysteine and actually extracting the sulfur if they need it. There are some which will use inorganic sources like sulfate and hydrogen sulfide as their source of sulfur. And the last absolute requirement is for phosphorus. Bacteria may also have a requirement for other things like potassium and things like that, but we won't talk about that. These are the major things, the major nutrients for them. Phosphorus, it occurs in the nucleic acids, DNA and RNA. It occurs in all of the nucleotides like ATP and all of the other nucleotides that we're going to talk about when we talk about DNA and RNA function. Next, you'll realize that the makeup of the membrane is made up of phospholipid. And the phospholipids consist of a phosphate group, that is the hydrophilic part, the hydrophilic head of phospholipid, and a lipid tail, the hydrophobic part. But those phosphate lipids together form this amphipathic molecule that goes to make up the membrane. For phosphate is usually, in most systems, is derived from some phosphate salt. For instance, we can use calcium phosphate as our source of phosphorus. And the calcium phosphate is the calcium salt, which is precipitated into bone. And in us, we use our bone as a source, as a store of calcium and of phosphorus. For prokaryotes, it has to be supplied in some way or another. But they're very good at breaking down sources of phosphate. We'll also see they may need various organic growth factors. It depends on the bacterium. Some of them manage perfectly well without and are able to synthesize everything they need. But some do require an input of an essential organic growth factor. Vitamins, sometimes an amino acid, purines, the pyrimidines like adenine, thymine, these sorts of things. They may require them and be unable to synthesize them themselves. It just depends on the bacterium. Then lastly, these trace elements, usually metal, which are associated often with enzyme structure. And the one that springs to mind immediately is hemoglobin. Hemoglobin is a complex protein of 4 different groups all joined together, and at the center of each of them, there is an iron atom. And that is why iron is one of the trace elements. But there are other metals which can also be required, like like copper, molybdenum, zinc, and even cobalt. Things like in very, 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 very small amounts. And that is because they're acting as enzyme cofactor. I tell you what I want you to do, just take a break for 1 minute. Okay, roll your head, stretch. We've done an hour and we've covered a lot of material. So just shake yourselves, have a little tiny break, give your poor brains a rest. Don't get on your phone. Just relax for a minute. And we'll carry on, okay? Any little break like this actually revives your brain. I think that I don't think there's a person in the world that can sustain concentration for more than an hour. In fact, I think our concentration begins to fade after about 30, 35 minutes. And our class is much longer than that. For these classes, the live classes, they are recorded and they will be posted for you as normal. So if you find that towards the end of the class you are losing concentration, I think just stick with it for the class, and then make sure that you go back and that you repeat. And I really advise you, when you use the video recordings, I really strongly advise you to make sure that you break it up into reasonable chunks. 20, 25 minutes, take a break, go through your slides. 20, 25 minutes more, stop, go through your slides, walk around the room, come back, sit down. Be kind to your brain. You can't stuff too much stuff in it, it'll start objecting. Okay, so let's just talk a bit about oxygen, because oxygen is so very important environmentally for prokaryotes. It's also a very important consideration in the laboratory. If we are dealing clinically with bacteria, it's very important that we understand what their oxygen requirements are, because they're quite varied. So for this diagram here, this is a little experiment that they have done. And we could do this in the lab if we, and all that you have to do is take a medium. This is nutrient rich. It's probably an agar, what we would call a sloppy agar. It's not a very high agar concentration. It's quite liquid, but not completely liquid. And in addition, inside we include a chemical. We usually use a chemical called sodium thioglycolate, which will it will bind all the oxygen present. If we just take a tube without, we don't inoculate the tube with bacteria at all. If we just take an ordinary tube and we set up a thioglycolate tube like that, we just leave it. We don't shake it or anything. After a while, what we find is when we go there, we would find that there's a gradient of oxygen. There would be oxygen up here at the top, much less here, then almost none, and down here, absolutely no oxygen. So we would set up an oxygen gradient. So now, assuming that that oxygen gradient is present throughout all of these tubes, we're going to inoculate these tubes with different bacteria. So here is one. We inoculated with bacterium, and then we left it for some days, probably put it at 37 degrees Celsius. It's a mesophile, so it grows happily at 37. We come back and we see where is this agar or this broth? Where is it turbid? Where is there visible evidence that growth is taking place? Well, in this tube here, there's where the growth is. Right at the very top. There's no growth down here at all. We can be pretty sure that this is an aerobe. It is an obligate aerobe. It is a bacterium which absolutely requires the presence of oxygen to grow properly. It will not grow in the absence of oxygen. In fact, it won't even grow in the where the oxygen is poor. It needs rich supply of oxygen. It is an obligate aerobe. And there are many of them. You'll commonly encounter them. The one that you will hear about again and again and again is Pseudomonas. And Pseudomonas is an obligate aerobe. Here's our second tube. This is a different bacterium. And what we see is that growth has occurred throughout the tube. It's even occurred right down here where there's absolutely no oxygen. It's occurred throughout the tube, but we see it's occurred a lot right at the very top. This is typical for an organism which is a facultative anaerobe. Anaerobe grows without oxygen. Aerobe grows with oxygen. Anaerobe, it grows in the absence of oxygen, but this is facultative. It will grow in the presence or the absence of oxygen. It's a facultative anaerobe, but there's something interesting here. Look at the growth at the top here, and you'll see the growth is very, very much stronger at the top than it is down here. This is typical for facultative anaerobes. They will grow in the absence of oxygen, but they grow much better when oxygen is present. And that stands to reason. Just think back to what we said in metabolism. We said at the very start of metabolism, we have glycolysis. It only produces a net of 2 ATPs. That is enough to keep things growing, but if you're only dependent on that little bit of ATP, your metabolism is going to go very slowly. You're going to grow much slower than an organism which can go all the way through and use oxygen as a terminal electron acceptor and run everything through cellular respiration. So this is reflected here. This is a facultative anaerobe. It can grow in the presence or the absence of oxygen. In the presence of oxygen, it's using cellular respiration. Much more efficient, much, much, much more efficient. And it's able to grow rapidly. But take away the oxygen and it can manage. It can carry on growing. It's just that it grows much more slowly. Here's our third tube, a third kind of bacterium. And here we see that there is no growth at all. There's absolutely no growth. If there's a molecule of oxygen present, this bacterium is not growing. And this is an obligate anaerobe. And it grows only in the absolute absence of oxygen down there. The next one, don't worry about. Aerotolerant anaerobes, we are not going to think about. I just want to come back here for 1 minute and tell you. It goes into a lot of detail here, which you don't need all of the detail. But what I do want to point out, one of the reasons this organism here will not grow if there's any oxygen present at all is it has no way to resist the incredible power, the oxidative power of the oxygen molecule. Oxygen molecules and various species of oxygen that are produced just naturally when oxygen atoms start smacking into one another. For whatever reason, they can produce these very reactive species of oxygen. And these obligate anaerobes have no way to deal with that. It very rapidly kills them. So they are limited to areas where there's absolutely no oxygen. These ones here and the aerobes and the facultative anaerobes, they have quite sophisticated means of dealing with those reactive oxygen species to ensure that they do the least possible damage to the cell. Now, the last group are these here. There are some bacteria which need a little bit of oxygen present, but they don't like a lot of oxygen. And again, it's one of the reasons is they can't deal with some of the oxygen species, reactive oxygen species that are present, but they do use oxygen as a terminal electron acceptor, and they run their cellular respiration slowly. And so they only need a little bit of oxygen, and they'll typically grow here in the middle of such a tube where there's a small amount of oxygen present, but not a lot. And those are the microaerophiles. Microaerophiles, we don't often encounter them in sort of in clinical lab, but environmentally, they're very important and very difficult to deal with. They tend to be what we refer to as fastidious organisms. They're quite difficult to grow. Okay, so I think it's now 10 past 5, and I really think that is probably enough for the day, and we'll carry on with probably complete this discussion at our next class. Okay. Remember that tomorrow I will put up a new assignment, and I have forgotten when your next test is, but I will let you know. I'll send a reminder for your test. I believe it's the 21st, somewhere around there. and that counts as your midterm. The last thing is, don't forget, 1 o'clock tomorrow. If you have specific questions and you want to ask me about anything, I will send you the meeting ID, and we can. I will see you on Zoom. Okay, and I think that is just about that for the day. Thank you.
Thank you, Professor.
Fine. Any questions before you go?
Professor, I do have one question, right?
Yes.
Looking at the syllabus, it stated that we're going to have the midterm test. That's on the 21st, but we're also kind of ahead of schedule as well for growth.
Yes.
We already started it this week. We technically started on the 14th instead. So that means that our midterm is going to only be on growth and genetics for the midterm. Did I confuse you?
No, it usually that second test is up to the class before the class before. That's a Wednesday, right?
For the midterm, yes.
The 21st.
The 21st.
21st. Okay, it'll probably. I think the midterm will probably be up till the previous Wednesday.
Okay, because I'm like, we're kind of ahead of schedule as well.
Yes, we are. We are a bit ahead of schedule. Yes, we are. The thing is, though, first of all, I do want to kind of slow down a little bit, but also it's just we cover a lot of material in a class, and it's a bit much to expect you to study everything from a Monday when you're writing the test on the Wednesday. It means you've only got a day to study a whole class, which is really not enough, right? So I rather make it up to the previous Wednesday to examine on that test, right?
Okay, no problem. Just wondering.
Good, and I will discuss it all in email anyway before the time.
Okay, no problem. Thank you.
Sure. Anyone else?
Sorry, I just wanted to ask if
go
it is.
ahead.
Oh, sorry. If it is possible for you to unlock our quiz answers, so we can see our answers and which are marked wrong, so we can see what we did wrong.
I thought I did. I'll check that immediately now.
Okay, thank you so much.
I thought I had unlocked it. All right then. Thanks.
Thank you.
Thank you, Professor.
Anyone else?
Do we have class on Columbus Day? No, right?
Oh no, you don't have class on Columbus Day.
Okay.
There's no Indigenous. I call it Indigenous Peoples Day. There's no class on a Monday. That's the 12th.
Sounds good. Thank you.
It's a holiday. Yep. All right.
Thank you.